Welcome back, friends. In the previous video discussion, we have talked about the introduction about the eukaryotic transcription process and how the eukaryotic transcription takes place and what are the important components for the eukaryotic transcription. Now, in this particular video, we'll be talking about a very important ingredient of eukaryotic transcription uh, for the ribosomal RNA. Two big subunit of the ribosomal RNA, that means the 28S and 18S ribosomal RNA, which is called the polymerase I or RNA polymerase I. Though the RNA polymerase I and RNA polymerase III are not that much common inside the eukaryotic cell, but still we are going to study them first. Then we will be talking about the major RNA polymerase or the RNA polymerase II, which is responsible for producing all the mRNAs which are helping to produce all the necessary proteins inside the cell. Okay, so we know that this is the polymerase I coding for producing 28S and 18S rRNA. Now this polymerase I, in each of these lectures we are going to see the structure of the DNA and the sequence of the DNA and the region inside the DNA where activators or mediators are going to bind, and also the regions where the polymers are going to attach. So it will form a total transcription complex along with the DNA, the proteins, the activator proteins or the mediator proteins, and RNA polymerase. So along with all these things, it will form a transcription complex. Now this transcription complex will allow to produce mRNA, to produce not only mRNA but different RNAs. Okay, so let's begin with polymerase I. Now RNA polymerase I, let me tell you something very important about this. RNA polymerase I is transcribing the DNA sequence, and for this transcription, so the activity of RNA polymerase I is simple or similar like the activity of Pol II and Pol III. So there are not that much difference in the activity between polymerase I, polymerase II and polymerase III in that sense, but the difference is in the mechanism of the transcription. So what it means is that the interaction pattern of this polymerase along with the DNA sequence, and also the interaction pattern of this polymerase with the DNA as well as the transcription factors will vary from one type of polymerase to other type of polymerase. For example, here polymerase I, so we are going to see polymerase I mediated process. Now in this process we will be seeing the DNA sequence structure and how it is recognized by the transcription factors, and then we'll see the addition of polymerase I and then the transcription process. Now in each kind of transcription you must know, this is a very important region. Before talking into this part, you must understand the basic process of DNA or RNA polymerization, and as well as you must understand the transcription process in case of prokaryotic system, because during this video I often will tell you that this is resembling the prokaryotic transcription initiation, this is resembling the prokaryotic transcription elongation, this is resembling the termination. You have to whenever I am telling you these terminologies, you have to catch these terminologies. So you must learn these terminologies first. You must learn a prokaryotic transcription in the basic level for the first time, then you will come back and see this video. Otherwise you can't be able to understand this. You won't be able, right? Why? Because I'm telling you the basic process of a transcription is divided into three different parts. One is the initiation, so let me write it here. One is the initiation, second is the elongation, and third one is termination or the ending. Okay, so in each different type of transcription is divided into three different parts, whether it is for the prokaryotic system or is for the eukaryotic systems. Now in case of the prokaryotes and eukaryotes, we can find remarkable similarities. Remember, remarkable similarities between elongation stage as well as the termination stage. There are a lot of similarities there, but the thing differ is in the initiation stage. So in case of eukaryotic transcription, the initiation stage is a hell and heaven difference than from the prokaryotic initiation, because in case of prokaryotes the initiation just begins with the signal recognition via the RNA polymerase itself, because the RNA polymerase for prokaryotes is a multimeric protein. It consists all the smaller subunits which are responsible for doing different things in a substrate, in an enzymatic form. So it's one in all. But in case of eukaryotic transcription, this polymerase 1, 2 and C are not just all in one activity. So they are having activity of polymerization. So this RNA Pol I, RNA Pol II or RNA Pol III in case of these eukaryotic transcription factors, eukaryotic transcriptions, they are just having the ability to polymerize. That means if we give them nucleotide sequences, so if we give them, say these are the nucleotide sequences, sorry, we won't give T, we will give U, like that, they will combine these two nucleotide sequences to join together to form the mRNA. This is called the polymerization. So they can only do the polymerization, but rest of the part, that means cutting the DNA, cutting the RNA, then joining it, then cutting the DNA, then joining it, then cleaving the DNA and unwinding the DNA, all of these things must be done with the help of special factors. Those are called the transcription factors. Now some of them are called specialized because they will be involved in special cases, and most of them are called the generalized transcription factors or GTF. Remember this term, generalized transcription factor or GTF. We must have access to this most of the time. Now these GTFs or generalized transcription factors in turn will be enabling this polymerase to do the function just like the prokaryotic RNA polymerase, which can function all in one. Okay, now in this case we are going to see the sequence, and there are specific regions in the DNA where these factors are having higher affinity to bind with. So there are stretch of DNA sequences are there. These proteins, these factors, these transcription factors having a higher ability, higher affinity to bind with those sequences, and they will bind with this sequence and they'll modify this and tell the DNA to be modified and tell this polymerase to be attached with, and then it will trigger the process in fast amount. So in case of all this case, the elongation process, which is simply made with the polymerization. So this elongation stage simply means the polymerization or the long lengthening up of the mRNA or tRNA or RNA, whatever. So it's a lengthening process of RNA. This process is similar to prokaryotes and eukaryotes. And then termination, this is differing for prokaryotes to eukaryotes. Initiation is also differing for prokaryotes to eukaryotes. So these two things differing for prokaryotes to eukaryotes, because this initiation stage is totally different than that of prokaryote and eukaryote, and this termination stage is also different from prokaryotes to eukaryotes, because this termination for eukaryotes again involves many more transcription factors and many more different types of proteins. They will in turn modify the producing RNA, modify the newly produced RNA. Modifications like polyadenylation at the 3' end and also 5' capping, and all these things must be carried out to finally produce and trim out the mRNA, which is ready to be translated into proteins. Okay, so these are the things I must tell. Then let us talk about this Pol I mediated process, and Pol I will produce this rRNA factors. Okay, now in case of this Pol I mediated process, let us consider a DNA, and this DNA, so if we consider this is a DNA, there are sequences. Remember, these transcription factors having affinity towards the particular sequences. Now in this case, those sequences, most of the cases, those sequences are called promoter sequences. Let me write it here. They are called promoter sequences. Now in case of this Pol I mediated process, the promoter sequences are consisting of two different parts. One is called, so let me write it here. So one is called upstream control element. One is called actual promoter. Another one is called upstream control element or UCE, upstream control element, or UCE, and another, the first one is the actual promoter. So if this is the DNA, we can find somewhere here the actual promoter. This is the actual promoter region. Suppose this is in between somewhere here. This is the start point. So here is plus 10, plus 20, plus 30 nucleotides are going in this direction, and here it is upstream region. So minus 10, minus 20, somewhere like this. So it's a minus, say, 110, minus 120, somewhere like that. From this region there is upstream control elements, or UCE. So this is, so let me draw it with a different color. I am taking green for that. This is upstream control element. So it is consisting of two parts, the actual promoter and the upstream control element. Okay, so actual promoter may come till, say, minus 60. This is whole actual promoter. So it is large sequence. Okay, so what we are looking at here, we are looking at this sequence. This is called the promoter region for this DNA for polymerase I mediated, RNA polymerase I mediated transcription process. So this is important. Two important regions. One is the actual promoter, where, remember, what is the promoter sequence? The actual region where the polymerase is going to bind. So this polymerase is going to bind with this actual promoter region. This is a large sequence. And further upstream of that, we are having the upstream control sequence, or upstream control element. Now this upstream control element is the region where transcription factors are tend to bind. Remember, we have talked about two different ingredients. One is this three different ingredients. One is the DNA sequence, second one is the RNA polymerase. Here is the RNA polymerase I, and third one is the transcription factor. Here again is a generalized transcription factor, or GTF. Now here, say this is a GTF. So let me draw GTF here. So with red color, let us draw it with red color. So we are having generalized transcription factors, GTFs. The GTF is going to bind with this UCE, or upstream control element, and then they are going to modify this, and they are going to recruit this RNA polymerase I and attach the RNA polymerase I with this DNA sequence and hold on to this promoter region. Now as this RNA polymerase I is fixed to this promoter region, it triggers the melting of the double strand of the DNA. Now as the double strand of the DNA starts to melt, it will allow new nucleotide sequences to be added and to form the mRNA chain via the elongation process. And remember, the elongation process for prokaryotes and eukaryotes resembles almost similarity. So this, I am not going to talk about this elongation, because I have already talked about the elongation when we have discussed about prokaryotic transcription. So just go back to those videos and watch them. But this process is similar one. But what differs is this initiation and termination. So I am talking about this initiation, which is the most important of all. So the initiation and the control of the initiation is a major thing for all these eukaryotic transcription events, where we require those transcription factors as well as the RNA polymerase. So here the generalized transcription factors are of different type. So one of them is called the upstream binding. One of them is upstream binding factor, UBF, upstream binding factor. Another one is SL. It is called SL1. These two things are among this GTF, or generalized transcription factor. There are different names for this generalized transcription factors. I won't encourage you to memorize all these names, but if you can, it's fine. But if you don't worry, but try to understand the concept. Now here, this upstream binding factor, as the name suggests, it will come and attach to this upstream control elements. It will bind with those, and this SL1 regions will also come and join them to produce a layer like structure. So what will happen? So again, let us draw the structure like that. This is the promoter region, actual promoter region, and this is, say, the upstream control element. So in this upstream control element, UBF start to attach. Say these are the UBFs. UBFs are attached. Along with them, SL1, say I draw SL1 like that. SL1, let us draw it like that. SL1, here say SL1. Now this is UBF1, not 1, UBF. Okay. So this is the DNA. The very first thing that happens is UBF will scan through and it will bind to the upstream control elements, and also they sit onto the promoter region. Now this UBF, remember this point. First, this UBF is going to bind with this promoter, bind with this upstream control element. Then it will recruit SL1 proteins. This SL1 proteins along with this UBF will have higher affinity to bind with the actual promoter region. So this SL1 protein is acting as the marker for the place. So it's a placement marker here. This SL1, as soon as it binds with this SL1, the UBF SL1 complex will have the destination to bind with this promoter sequence. Remember, this is the promoter sequence. And we know from our basic concept of transcription, when a polymerase is going to attach with the promoter, it will start transcription. So this transcription factors SL1 is an addressing molecule. Remember this term, addressing molecule, because it is addressing polymerase I to be attached with the promoter. So I have to erase this. No, besides there. Anyways, so it is called the addressing molecule because whenever we are having the complex of UBF and SL1, it is addressing, it is marking this promoter to bind with polymerase. So it will bring polymerase I to the scene. So here is a polymerase I sitting here. It will bring polymerase to the scene, and then what will happen? It will happen something like that. So we are having this DNA. I'm not going to draw in detail. So this is the portion. Sorry, this is the control element, and here we are going to see these UBFs. So again, we have two UBFs here, and we are having the SL1s like that, and then this polymerase will come and say this is the RNA polymerase. Okay, so this large molecule is RNA polymerase. So it will bind with this promoter region. So what is this? Remember, this is the promoter region. Into this promoter region, Pol II, RNA Pol II bound. Now RNA Pol II, as soon as this RNA Pol II bind with it, it will start to modify this DNA structure. It will trigger the DNA structure to be melted. The DNA double strand is melted, and then it will allow one of the DNA strand to be the template strand for the synthesis of mRNA. Because remember, in case of this RNA synthesis, one of the DNA strands act like a template strand. So it will act like a template. It will allow the nucleotide sequences to come, and then elongation process will start. I'm not going to talk elongation in detail. Then the whole elongation process will go on, and then finally termination will occur. In case of prokaryotes, we can see the termination is happening in two different ways. One is a row independent, another one is a row dependent fashion. But in this case, we won't see those kind of termination. Instead, we will see the presence of some other type of proteins that will form a complex like structure. Then it will fall out or take this mRNA out of this place. Okay, but here in this video I'm not going to tell about the detail of this termination, because we will be seeing this termination process in detail in our later videos. Now here is the process sitting on to this, but remember the concept. Don't try to mug up all these things, just remember the concept. We are having transcription factors, but the actual goal of the transcription factor for polymerase I mediated transcription, as well as we are going to see the polymerase III mediated transcription, is that they are having the transcription factors. One of them or few of them is going to bind with the upstream elements which are acting like a mediators or the activators of the process. Some of them are going to bind with those activator regions of the upstream elements. Then rest of the part are going to attach with the promoter region or actual promoter region. So those second part is called the addressing molecule, because those addressing molecule will give the room for other molecules to go and bind with the actual promoter region, and they will recruit the polymerase to the place, and then the whole process will start again. Okay, so this is a process of polymerase I mediated transcription, and I hope this will help you. Thank you.